Hello, I'm Tom Riffelmacher, and I'm here in the lab just recording a quick message for the Rickies. I know you missed the Spark event, so I wanted to just convey the words that I was also targeting to you in a direct message. Briefly mentioning what the Spark program did for me. So when I joined Mitch Kronenberg's lab more than 5 years ago now, it was quite humbling to me. He's a very famous scientist. He's incredibly good. He knows incredibly much about immunology, and I really didn't. So I felt like for the first probably at least 2 years, really the best thing I could do was following his advice, and that led to good success and was a recipe for success for sure. However, I think because thanks to the Spark program, when I was asked to actually develop my own ideas, I kind of discovered that, I can also try and test some of my own ideas, and some of them are not too bad actually. Some of them actually worked out incredibly well, and I'm sitting here in the room that has the seahorse instrument. This was one of the instruments that was crucial for the study that I did on metabolism, and it was kind of a follow-up, but in an indirect way, on that Spark project, asking about hypoxia and oxygen in how T cells respond. And some of this work, I'm really happy to say, we just recently published in Nature Cell Biology, so a very, very prestigious journal. I'm very proud of this work, and I think the Spark program had not just a little role in this, so by extension that is also owed to Spark. Now, one of the coolest data that we got in that project, I just want to show this very briefly. Let me see if I can change the camera. Don't know if you can see this, but this is one of the key figures, and this is one of the key experiments we did, looking at fatty acid uptake. What we found is that this one subset of T cells took up huge amounts of fatty acids, whereas the other subset of T cells didn't take up any fatty acid at all. And this really has functional implications. These guys, they need that to have an effector function producing IL-17, so a really important cytokine that is actually driving some fat-related diseases. So here we have a T cell that is highly dependent on fat uptake, and it's producing a cytokine that is very important for fatty liver disease and for some other diseases that are related to too much fat basically. So really interesting connection between metabolism and T cell function and disease. And this is something I'm quite excited about, and that I'm hoping to explore next. I'm actually writing a R01 grant. I'm in the final stages of that. This is one of those huge career making or breaking grants. We're applying for a total of $2.3 million over 5 years, and so this is very crucial. And I'm hoping that this will allow me to get some of the preliminary data to get a grant like this. So again, Spark is just so important for our research, and thank you for supporting it. And that's a short update from me from the lab. So thank you, and bye bye.